My name is Scott White, not Mike Cola, and I'm here to talk about PanV and our pan-antiviral antibody strategy targeting viral infections. First, I want to talk about the team. The original discoveries for PanV were made in the laboratory of Dr. Akiko Iwasaki, whose exceptional reputation and really, I think, the gravity of these findings have attracted what I see as a very serious development team that includes Mike Cola, who's got over 30 years of industry leadership, and his experience includes spinning out technology from several academic institutions, and scientific advisors that include myself, Drs. Cookland and Pierce, each of us with over 20 years of large pharma and biotech experience and expertise that ranges infectious diseases, oncology, immunology, and antibody development and production. I don't think I need to say anything. I think we all recognize our global vulnerability to viral infections and outbreaks, whether it's new viruses or newly emerging viruses. But what PanV's technology has to offer could really reshape the landscape in that it avoids targeting peptide epitopes, which are susceptible to viral mutation, and goes after what has been discovered as a carbohydrate moiety that's actually placed by the host cell protein synthesis machinery. The original discovery was made of a naturally occurring innate monoclonal antibody that binds to endogenous retroviruses and seems to play a role in preventing their reemergence. And you look more closely, what you find is that apparently the architecture of these envelope glycoproteins restricts glycosylation through the host cell machinery for protein synthesis and reveals a distinct epitope on the virus that isn't found on host cell proteins. And actually, if you go and look at other viruses that also have surface glycoproteins, you find that PanV1, this monoclonal antibody, recognizes these glycoproteins, whether they're in isolation or in the context of the intact viral particle, and again, from a wide variety of viral families. Exactly. So this finding, if it can be brought through to a product, really has a chance to reshape the landscape for the treatment of viral infections through a single agent that could target a broad range of viruses in a way that depends on an antigen that's placed by the host glycosylation machinery. What this also does is open up several potential partnership opportunities based on interests in different types of infections, whether that be respiratory pathogens, vector-borne illnesses, emergence of reactivation of infections in immunosuppressed patients, or biothreat pathogens. And over the last year, with Biotech funding, we've been able to produce a humanized version of the antibody and better understand the nature of the original monoclonal binding to its target. What we're doing over the next several months is optimizing that antibody candidate to develop a series of lead and follow-on candidates that we would like to take into further development. We currently have a commitment of $5 million of equity investment from the Gates Foundation after having seen this information, and what we're seeking now is a co-lead for Series A funding to continue the development of the asset. And that's all we have. Thank you very much.